And thanks for the invitation to join the EMSL Summer School. I'm really excited to be here today, and also to thank the entire team at EMSL for having the National Microbiome Data Collaborative team join on this summer school effort over this week. So I think, as you heard from John and Maggie, there's some really exciting efforts as part of Monet and the Thousand Soils Project. And what I'd next like to talk about is how we're going to be able to support a lot of that data that's being generated. And so for those of you who may not be familiar with the NMDC, we are a pilot program. We got kicked off in July of 2019. We're funded from the Department of Energy Office of Science. And I have just a high-level overview to share, and you'll be hearing from others over the course of this week and getting some hands-on experience during the afternoon sessions for using a lot of the NMDC resources. So with that, I'll just get started and share kind of why the NMDC exists and what we're trying to accomplish. So for those of you who are either new to the field or have been in the field of microbial ecology for a while, hopefully you can appreciate that the field is just exploding with the amount of different types of omics data that are being generated, different tools and techniques for generating data on microbial communities. And it's a really exciting time to be part of the field. And one of the aspects that we've thought a lot about over the past several years is how best can we support this growing volume of data, and how can we support it in a way that will enable researchers across the globe to be able to analyze and interpret their data within the context of other studies and data. And so this is a graph that I like to show that illustrates really the problem that we're facing. So if we think about in the green cube here, that this is kind of the current state of analysis. So currently you're able to look at the taxonomic, the ecological, and the genetic scale within this kind of defined area with the amount of data that's already been generated. But if we take a look out into 5 or 10 years from now, what is that data landscape going to look like? And that's really illustrated here in this much larger yellow cube. And so this is all to say that all of the great advances in DNA sequencing and other omics technologies has outpaced our current data infrastructure. And another way to look at this is we have many nice resources currently available that have been developed over the past several years or even decades for how to manage and handle sequencing and omics data. But if you try to go from one resource to the other, it's really challenging to do so. And the reason for that is that these resources have developed, for the most part in isolation, and have not purposefully tried to coordinate and harmonize data across systems. And so what we would like to do as part of the NMDC is to move towards a system more like this, where you can easily share data across systems. You can identify data from one system that might be available in another, and that you don't spend all of your time trying to reformat data and really work with data, and that you can really focus on doing the science that you would like to do and be able to look at new and interesting questions with all of the compiled data that's been generated to date. And so that's really the reason that the NMDC exists. And imagine if you didn't have to spend all of your time wrangling data and instead trying to answer really important grand challenge science questions like you heard from John and Maggie, particularly about the Monet project. So just imagine the possibilities. So these are a couple questions from researchers that we've worked with over the past 3 years, and trying to think about if they could analyze their data in the context of their own study, as well as doing comparative analyses across studies. Can they look at the role that microbe plays in the persistence of soil carbon? Can we look at questions of how microbes and viruses mediate these watershed nitrogen transformations at scale, or how microbes impact biogeochemical cycling in deep subsurface environments? So these are really important grand scale challenges that the NMDC is really designed to try to help researchers support and tackle. So we're really looking at new ways for researchers to ask questions with data, and importantly, to have robust integration across samples and scales. And so what we did several years ago now was to initiate the concept of the NMDC. And so I just wanted to spend a couple minutes walking through some of the history of the program so you can have an understanding of what it really took to spin up this pilot program and what we're hoping to accomplish as we transition from this pilot phase into a more production scale system. So this all really started back in 2017 at a workshop at the Joint Genome Institute. This is a picture back at our old Walnut Creek site. And we really were focused on convening some of the thought leaders in the field of microbial ecology and metagenomics to discuss what the needs were within the community to support collaborative data infrastructure. This was led by Nikos Kirpidis, shown here in the back, and myself, along with Claire Fraser, shown here, and Ed DeLong. And what happened after this workshop is that we focused on a lot of follow-on activities. We had a town hall at the ASM Microbe meeting to really share out with the research community what was discussed during the workshop and how we can collaboratively as a research community work towards what would be needed for this larger data infrastructure. We also had a stakeholder workshop at ASM where we brought together researchers, infrastructure providers, as well as funders. So funders not just in the Department of Energy, but at NSF, NIH, and other main federal agencies that fund microbiome research to really emphasize the need for aligning the community and how to best support data as the community develops and does these much larger grand challenge projects such as Monet. And then there was also, importantly, a congressional hearing from our Berkeley Lab Associate Laboratory Director, Mary Maxon, to really emphasize at the congressional level the value of data infrastructure for microbiome research. And all of this worked together to emphasized the need, and we were asked by the Department of Energy to lead a multi-lab national effort to develop this community-centric NMDC framework back in 2019, as I mentioned. And so this is an overview of the current NMDC vision. So we're really aimed at empowering the research community to harness microbiome data exploration through this integrative data ecosystem. We're led out of Berkeley Lab, and we include team members at Los Alamos, Oak Ridge National Lab, and Pacific Northwest. And the focus of the NMDC is on these circles on the outside, focusing on data standards, which I'll talk about in a few minutes, data quality, data integration, so bringing together multi-omics data, and importantly, data access, so providing data in an accessible manner and providing it in a way that researchers can come to the NMDC, get the data that they need to enable their research. And so I wanted to step through a couple of the outputs that the NMDC team has worked on over the past 3 years. And so we're calling this the NMDC's infrastructure backbone. And it really focuses on 3 main outputs that our team has worked on. The first that you'll get some in-depth hands-on work over the next couple days is the NMDC submission portal. And this is a screenshot showing what that portal looks like. And the focus of this portal is to really lower barriers to collect study and biosample information. So you've already heard about the importance of metadata, the value of understanding all of the great environmental and contextual information from which a sample comes from. Well, this system is really focused on trying to make it easier for researchers to provide that information in a more systematic way. And so you'll hear a lot more from Montana about this later this afternoon. The second effort that the team has worked on is called NMDC Edge. And this is really focused on streamlining multi-omics data processing. So we have, as part of the NMDC, a suite of bioinformatic workflows that have been standardized and have been used in production at the user facilities, JGI and EMSL, for many years, using best practices for data analysis, and are made easily accessible and available through this user interface that, again, you'll get some hands-on work with Karen and the rest of the team over the course of this week to be able to analyze your data using this system. And then lastly, we have the NMDC data portal, which is one of our main products to get access to multi-omics data. And you'll hear a lot more over the course of this week about how to use it, some of the unique functionality, and how we've really focused on bringing together input from the research community to be able to inform the design and look and feel and features of the portal. And you'll hear a lot more about this from Pajal later today. And so in addition to building this infrastructure backbone, the team has also really focused on creating an engaged community. And what do I mean by that? We're building infrastructure not for the sake of building it and hoping users will come and access data and using resources, but really being closely tied to the community, having a good understanding of what the community needs, as well as building strong partnerships to have a more sustainable approach for how we develop as a research community going forward. So a couple examples of what that looks like. Our team is heavily engaged and involved with the Genomic Standards Consortium. For those of you who are familiar with metagenomics and genomics data, the Genomic Standards Consortium is really the go-to resource and group that handles standards for sequencing data. So that can include anything from how that data is identified and available through primary repositories, ensuring that minimum standards are used so that when data is deposited in those primary repositories, researchers understand where it came from and some of the main metadata components as part of that. Another example is we have a very long-standing relationship with the American Society for Microbiology. Through partnering with this very large society, we're able to do some exciting activities, such as launching the ASM Microbiome Data Prize last year to recognize the value and importance of the intersection of computing science and microbiome research. So those are just a couple examples of how the NMDC is working on fostering partnerships. And also we're very interested in how we can support researchers more broadly. And so you'll hear about the NMDC Ambassador Program more later today, I believe, and how we're working with early career researchers who are interested in promoting standards and training opportunities across their various communities that they work in. And so these are, again, just ways that the NMDC is working across the research community to broadly support how we can support data science and microbiome research. All right, so as you've heard, we are working across the two DOE user facilities, the Joint Genome Institute and the Environmental Molecular Sciences Laboratory. And importantly, I wanted to highlight that we have this fantastic partnership because we're very strongly aligned with both of their strategic objectives across these user facilities. So on the JGI side, where I also sit, there's a strategic scientific thrust in microbiome data science, and the NMDC fits squarely within the strategic thrust to support standards, support bioinformatic data processing, and providing, as you've heard from John, how we can support fair data across the JGI and the NMDC. On the EMSL side, we have a fantastic partnership with being able to integrate multi-omics data that's generated at the EMSL facility and be able to, in the next phase of the NMDC, support data analytics and visualization in a meaningful way. And so what does this look like in terms of how data flows across facilities and resources? So the JGI and EMSL both have these user facility data portals that are pictured here. And what the NMDC does is able to access that data through APIs from those resources to integrate data from multi-omics data. So if you're a JGI or an EMSL user, you're probably familiar with the process of going to those data portals and accessing data through those systems. What the NMDC is doing is to pull data across those two user facilities and be able to provide it within our data portal for access and for search and discovery. Importantly, we're also emphasizing how we can partner and link to complementary resources. So examples of that include working with the ESS Dive team for hosting data that can be shared across the systems. We're working with the JGI IMG team, the Integrated Microbial Genomes and Microbiomes resource, to share data that has been processed at the JGI and is available within that comparative analysis platform. And you'll see some examples and be able to have some hands-on work to get a sense for how that looks. And then just this past spring, we're really excited to work with the KBase team, so the DOE Systems Biology Knowledge Base, or KBase, to have all of the data that is currently hosted on the NMDC for shotgun metagenome sequencing, have that available through the KBase interface through a KBase organization, so that users who are interested in doing more advanced analyses with the data, for example, metabolic modeling, can take that NMDC data and spin off a variety of different narratives for working with that data. And so I now wanted to get into some of the details for what the NMDC is doing in terms of metadata standards and how we're broadly supporting and working with the Genomic Standards Consortium. So for those of you who may be familiar with the GSC, and especially with the minimal information about any X sequence or the Mix-S standards, these are broad reporting standards that specify how information is required to be able to submit that data to a primary repository. And so for those of you who may be familiar, there is a large community that brought together the reporting standards that's listed here in the 2011 paper. But I think importantly that these standards are managed as Excel spreadsheets. And for those of you who have done large comparative analyses or are looking to identify data within NCBI or other primary repositories, this isn't a great way to have standards represented. They're not machine actionable, they don't follow the FAIR data principles, and they're not modular. And so what does this look like? So if you go to any of the INSDC resources, that includes NCBI, ENA, or DDBJ, and try to get information that would be consistent with those minimal standards, you can get information back such as this with depth. So these standards are not well represented. There's a lot of flexibility and ambiguous interpretations for how this data can be submitted. And what that results in is that the data really is very noisy. And so often people, researchers need to go and clean up that data and metadata before they can actually use it for their downstream analyses. Another example is looking at pH, and you can see all of the variety of ways that data has been deposited within these primary repositories that really just presents a whole lot of challenges for researchers who want to use this data and make meaningful interpretations of it. And so going into a bit more detail, I know this is very busy, but this is an actual snapshot of what one of the environmental packages looks like from the GSC. So there are many, many different metadata terms. For this particular example, there's 148 that describe all of the various aspects of a sample from a soil environment. But when it comes down to it, it really, if you were to look at this, Excel spreadsheet, there's a lot of ways that it's just very overwhelming and very difficult for someone, especially new to the field, to be able to really comply with the standards. And so over the past 3 years, this is input that our team has received about the GSC standards, that the templates have way too many terms. It's really overwhelming. It's difficult to find terms that are relevant for their particular study. They may not understand the definition. Where do you go for help? What's an ontology and why should you use it? And so these are, some important pieces that even though these standards have been around for almost 10 years now, that are really challenging researchers in how they can even comply with the standards and be able to use them in a meaningful way. And so over the course of the past 3 years, the NMDC has really focused on making these standards fair. So working very, very closely with the Genomic Standards Consortium, and particularly the Compliance and Interoperability Group, to take the MICS reporting standards in Excel and move them towards this updated version using a machine actionable web searchable standard called LinkML. And so this was work that was really spearheaded by Mark and Sujai on the team. And we can get into more depth about all of the work that went into making this transition from an Excel spreadsheet to the LinkML framework, but it's really an exciting accomplishment. And I think will really move the field forward in terms of how these standards can be more readily adopted across teams and can be updated going forward. And so one of the outputs from transitioning the standards into this LinkML framework is to provide this in the NMDC submission portal. And so this is a snapshot of the submission portal that uses the standards. And you'll hear a lot more from Montana later today about how to use this portal and how it will really enable researchers to provide metadata in a much more streamlined fashion and be able to do so that wasn't previously possible using the Excel spreadsheets from the GSC. All right, I now wanted to move towards why we spent all of this work in updating the standards to the LinkML framework and how that is a modular way for how we're building out the NMDC. And so importantly, we have focused on developing a schema for fair data that underpins all of the aspects of the NMDC. So this provides really a modular design for how we collect information on studies, biosamples, how we support the omics processing and standardized data outputs, and then how that is made available within the NMDC data portal. And so again, this is work spearheaded by Mark, Sujai, and Patrick from Chris Mungel's team at Berkeley Lab. And so, as I mentioned, that this schema really underpins all of the NMDC architecture. It's used to support the submission portal. It's used to provide information and data to the central NMDC metadata store. And then it really drives the faceted search that you'll get much more acquainted with over the course of this week. And then next, I wanted to move on to not only are we focused on standardized metadata, but we're also focused on supporting standardized bioinformatic workflows for data processing, such that you can not only query and find data that has similar metadata fields, but you can also search and identify data based on functional annotation features. And why is this important? This is important because if you want to compare your data that was collected in a particular way to data that was collected from a completely separate study, if you can't harmonize the processing of the data, that you just are not able to integrate those data types in a way for meaningful analysis. And so what has the NMDC done? Well, we've worked very closely, as I've already mentioned, with the JGI and EMSL to adopt the production quality bioinformatic workflows that these user facilities have developed over the past several years, if not decades, to be able to support those in ways that researchers can have access to those workflows and be able to use them to process their own data, even if it was generated outside of the two user facilities. So we have standardized workflows that span metagenomics, metatranscriptomics, metaproteomics, and a couple different flavors of metabolomics. And so the way that we're making these workflows broadly available outside of Docker containers, especially for users who may not be familiar with Docker containers or how to run those particular images and may not have the bioinformatics background, we have a user-friendly, accessible platform called NMDC Edge, which is shown here, to be able to have just point and click, upload your data and process it and get your outputs. And so you'll hear a lot more from Karen on day 2 for the hands-on session using NMDC Edge. We released a beta version several months ago, and this supports execution within a high-performance computing environment, and we have a lot of really nice documentation and tutorials on how to use the NMDC Edge platform. So just a snapshot of what workflows are currently available. So we've focused on providing the end-to-end metagenomics workflow. So that includes all of the production processing that JGI does, from read QC analysis to metagenome assembly, annotation, and metagenome binning. And then on top of that, we also have a reads-based analysis developed by the Los Alamos team to support further analysis of that data. We also have metatranscriptomics workflows and a natural organic matter workflow that, again, you'll have hands-on work with later on this week. All right, so I now wanted to jump to the NMDC data portal and how you can get access to all of this data within the NMDC portal system shown here. So we launched this back in March of 2021. We have integrated metadata. I guess this slide, sorry, is a bit out of date, but we have 8 studies that are integrated into the system. And we've leveraged distributed data resources at NERSC and at EMSL to provide this. For those of you who may not be familiar with computing architecture or infrastructure, that really probably doesn't mean a whole lot. But what, for those of you who might be familiar with it, it means that we're not a centralized computing resource, that all of the data is available within kind of a brick and mortar building. It's distributed, and that's a way that we can really build out the NMDC over the next several years to be able to scale to the vast amounts of data that are being generated, and for research teams who want to have their data made available within the data portal. As I mentioned, we have integrated multi-omics data across a number of studies, and we've done so harmonizing the metadata, but then also harmonizing how that data is processed through these standardized workflows that I just mentioned. And so what that enables is that you can not only search, shown here in the left-hand panel, search across different metadata terms, but you can actually search across different functional terms, which is shown here with the KBase term. And so what that enables, that if you're interested, say, in a particular gene of interest, you can search for that gene across metagenomic, metatranscriptomic, and metaproteomic data, which I think is a really powerful way to get access to data you might be interested in. And then we're also tracking data downloads in an effort to provide metrics and information both to researchers who provide data as well as to data users who are accessing that data. All right, so as I already mentioned, we have a nice partnership with the KBase team where you can go to KBase system and be able to develop a set of narratives to do further analyses off of NMDC data, which I think is a really exciting way to take the NMDC process data and do downstream analyses such as metabolic modeling. We also have a really nice partnership with the IMG team. And so data that you see within the NMDC data portal is the same data that you can access through IMG. And so the IMG team just recently launched all of the NMDC study information within, if you go to collaborations within the IMG interface, and you can get access to reads-based analysis, which augments all of the assembly-based metagenome data that IMG hosts. So again, this is a nice way that we're trying to not just build infrastructure that is new, but be able to integrate across existing infrastructure to be able to better support how data can be analyzed and interpreted across systems. All right, so I think I'm getting close to my last couple slides here. So I wanted to just highlight that the NMDC team has really focused a lot on developing this documentation framework to really support researchers to understand all of the various aspects of the work that the NMDC does. And so this was really spearheaded by Karen and Julia, shown down here. And it focuses on kind of these 4 different modes of learning and training. So it includes tutorials, how-to guides, explanation, and reference material. And so if you're interested to learn more about any of the aspects, if you want just a quick how-to guide, or if you really want to get in depth about any of the workflows, you can do so going to the link down here. All right, and this is my last slide. So hopefully I've covered kind of the main aspects of the NMDC and really the areas that we're focused on working in. And so we have a lot of material. We're an open science platform. So all of the work that we do is in GitHub. We have our data portal, which is accessible, as well as NMDC Edge. We have a variety of ways to get involved with the NMDC, whether it's through our newsletter, joining us on Twitter, or becoming an NMDC champion if you're really interested in the work that we do and becoming more actively engaged and participating in our program. And then here's a couple of the publications that we've had over the past couple of years. So with that, I'd be more than happy to take any questions. I'm really excited to, again, be a part of the EMSL Summer School and looking forward to a really exciting and productive week. So thanks so much.
Great. Thank you so much, Emily. So we've had a few questions come in as Emily was speaking, but again, if you have anything you'd like to ask, please check out the Discord and add your questions there, and we will funnel them. One of the first questions we got was from Andrew Lalu. He asked, can we submit data from other countries?
That's a great question. We are agnostic to where data comes from, even though we have that sticky national as part of our name. We are encouraging use of our submission portal, but we currently don't have the connections yet to offer when you submit metadata to the submission portal that goes to the data portal. Same with NMDC Edge. If you submit data to NMDC Edge, you can get it processed, but that doesn't eventually make its way to the data portal. So over the next year or two, we're really excited to build out those connections so that anyone who's interested in depositing their process data at the NMDC, that they can do so, but that capability is not currently available. However, if you're interested in using any of the resources, please do so, and then also get in touch with our team so that we can work with you on having that data made available through the data portal.
Thank you. All right. Dr. Abisk asked how much the amount of data can be processed using NMDC Edge. And I believe Karen is here. She might be able to help with that too, Emily.
Yeah, I'm going to punch